actually happened over at the New England Aquarium. My daughter and I were over there, and they have this display down there in the basement of these jellies, jellyfish, and they were swimming around like this. And I looked at it and I said, that's a muscular pump, just like the heart. The heart's a muscular pump. And I said, I bet I can build that. They're simple enough that I bet I can build it. And I bet I can build it out of cardiac muscle cells. So we took apart a rat, we rebuilt it as a jellyfish, we matched the swim behavior, it all worked the same. And then we had an idea that maybe we needed a more complex structure for undulatory motion, which is because the heart, when it beats, it squeezes from the bottom up. And so my daughter and I had gone back to the New England Aquarium. She was trying to touch a stingray. They have kind of a petting zoo there. And as the stingray came by, it kind of deflected, moved off like 45 degrees. And she looked up at me like, Daddy, what was that? And it hit me like, that's it. That fine motor control to turn like that is probably some of the architecture required to do that. It's probably similar to what we see in the heart. And so we did that. And we learned a lot about undulatory motion and the musculature required to achieve that.
My name is Gil-Yong Lee. I'm working on the cardiac tissue engineering in this lab.
Young came in and really kind of focused on this project, and it just showed an extraordinary discipline and attention to detail.
Biohybrids are interesting because, in my mind, they transcend this space between kind of living and artificial. And so they start to be this cyborg system where they're both, you kind of get into this area where if you ask, are they alive or are they dead? The answer is a little bit yes to both.
So with the fish that we're building right now, which is basically a little bit of a model like the zebrafish, the idea there is that you've got two pieces of muscle. And when the fish swims, it's wagging its back end back and forth, and that's how you get the motion.
When one of the muscles contracts, the other one is stretched. The stretching activates the mechanosensitive protein, thus leading to induced the muscle activation and contraction. This will repeat and create the fish-like motion.
That's exactly what we're doing with the heart. They go one direction, they contract in the other direction. Two different muscular layers to do that, we think, or multiple muscular layers to do that. We were trying to replicate that when we did the zebrafish. We knew that there was probably some unique biophysics that we could replicate or exploit to build a pacemaker. So we made this little thing called the G node. It's this little spot above the tissue that kind of tickles, and you get this wave of excitation that flows through the whole tissue. And so we have an autonomous pacemaker. So we no longer have to control this thing with light. This fish controls itself and can swim on its own because the excitation wavefronts come out of the geometric node.
Because of the two internal pacing mechanisms, our fish can live longer, move faster, and swim more efficiently. Our previous stingray could stay alive for only 7 days max, but our new fish can survive for more than 100 days. This fish swimming efficiency reaches the efficiency of the live real fish.
Human stem cell has the purity is higher than some lessers, which it can increase the longevity, and also we can apply to human cardiology.
The idea was that the fundamental building block of a muscular pump should be the same. In other words, I could build a fish with a heart cell. That was the first thing, right? So we built a jellyfish, we built a stingray out of rat cells. But now we wanted to take it up a notch because we wanted to start testing our ability to build muscular pumps to go into patients. We got to build them with human cells. So we had to start using that substrate. Start with the stem cell, evolve it to a cardiac myocyte, then take that cardiac myocyte and build the fish. So it was challenging in that now we started to have to battle cell biology or stem cell biology when we started using the human cells. But the great thing about this is that the work that's been done by Gil-Yong and son Jin is they can keep these cells, the human cells, alive in a dish for a long time. When they mature in the dish, they get stronger. so we can keep them longer in the dish. We can keep these fish swimming longer. And using cardiac muscle cells from the heart to replicate that fish, that gives me a great control experiment. And really what I'm doing is seeing how good am I at building muscular pumps. Before I try to go live with putting one of these things into a sick kid, I need to be pretty sure that I'm a good engineer, that my team is good. And that's why we build these fish. 안녕하세요. Biohybrid. Jahakyo, Gayoso, Kevin K. Parker, Ibnida. In Kyong, Shimkyong, Kyongyong, Sapola, Manjon, In Kyong, Mokoyok Li, Sokahago, Sipsunida. Do some bears. Do you think that was as good as the last one?
I think it was better.
No, man.